Good evening, everyone, and thank you for attending tonight's Research Tuesdays event, Taking on Type 2. My name is Dr. Nigel Rogash, and I'm a Senior Research Fellow here at the University of Adelaide and your MC for tonight. I'd like to start by acknowledging the Kaurna people, the original custodians of the Adelaide Plains and the land on which the University of Adelaide campuses at North Terrace, Waite and Roseworthy are built. Tonight marks our third Research Tuesdays for 2021, where you'll hear from an incredible speaker who was awarded the university's 3-minute thesis overall winner in the categories for 2020, Miss Kalia Primer. The presentation tonight is a continuation from Kalia's 3-minute thesis win on type 2 diabetes and its vascular complications, including heart attack, peripheral artery disease and chronic wounds. One area of her work focuses in on how blood vessels break down blood sugar for energy and how diabetes impairs this. Vascular complications are the major cause of death for people with diabetes, so understanding the pathology involved and identifying new therapeutic targets is essential to protect patients from these conditions. Kalia will deliver a presentation followed by a Q&A session with our audience here in Bragg's Lecture Theatre and also online. Thank you to those who have submitted a question with your registration. We will answer some of those along the way. I also encourage all our viewers online tonight to continue to submit questions during the presentation via the Q&A section down the bottom of your screen. Now I'd like to officially welcome your speaker for tonight. Kalia's PhD's research focuses on diabetic vascular complications and on understanding how the metabolism of blood vessel cells affects their ability to undergo new growth. When not in the lab, Kalia volunteers with a number of committees dedicated to supporting medical research in South Australia, including as convener of the SA branch of the Australian Society for Medical Research. Please join me in welcoming Kalia Primer.
Thank you very much, Nigel. Just before I get started, if I speak at this volume, can everyone at the back hear me okay? Is that all right? All right, cool. Seeing some thumbs up. That's great. All right, and also before I begin, I would like to say a huge thank you to Liz and the rest of the Research Tuesdays team for inviting me to speak tonight. It's not often that I get an opportunity to speak about my research with such a large audience, ever actually. So I'm really excited to be here, and thank you all for coming along to listen. So as Nigel said, my name is Kalia and I'm a PhD candidate at the Adelaide Medical School, but I'm actually physically based, and I'll just see, can you see my mouse? You can, okay, cool. I'm actually physically based at the South Australian Health and Medical Research Institute, which is all the way up the other end of North Terrace. We call it the SAMRI. My supervisor's group is based within the Vascular Research Centre, and that's within the lifelong health theme at SAMRI. So SAMRI sort of contains about 4 major research themes, and this lifelong health theme really focuses on understanding and doing research into diseases that affect us all across our lifetime, including things like diabetes and cardiovascular disease, but also Alzheimer's and diseases like that. I do have to add this little disclaimer here, which is that I am not a clinician, I'm not a health professional, which means that I really can't answer questions or give advice about the clinical management of diabetes. So I'm very sorry to disappoint if anyone had a planned question in that area. But there are some things that I can definitely talk about. So this is an overview of what I'm going to discuss tonight. Firstly, what is diabetes? How does diabetes affect our blood vessels? Why is new blood vessel growth so important? And then at the end, I'm going to discuss some new ways that we're working on in order to help blood vessels grow and function in diabetes. So firstly, what is diabetes? Well, firstly, we want to talk about blood sugar or blood glucose. This is your cell's main source of energy, and it comes from the food you eat. So once you've had a big meal, all the nutrients get absorbed from your gut into your bloodstream, and glucose makes up a significant portion of those nutrients. Another thing that happens once you've had a big meal is that your body, or your pancreas specifically, starts producing insulin. And insulin is a hormone that helps glucose get into your cells so that they can break it down and use it up for energy. However, diabetes is, the hallmark of diabetes is really that your blood glucose or your blood sugar is too high for some reason, and that really comes back down to what insulin is doing. There's two types of diabetes, type 1 and type 2, and I'm mainly going to talk about type 2, but I want to just quickly discuss the differences between the two because it's really quite interesting. And a good way to think about the differences between type 1 and type 2 diabetes is to imagine insulin as a key. If we say insulin is a key, it unlocks the door to the cells that lets the glucose in. So in type 1 diabetes, there is no key. Your body doesn't produce insulin properly at all. And type 1 diabetes generally onsets much earlier, usually before the age of 40. And it's predominantly, we believe, to be a genetic condition with some environmental factors involved as well. This is in contrast to type 2 diabetes, and there's sort of two aspects to this, which is when the key is there, so your body is producing insulin, but it either doesn't unlock the door properly, so the insulin doesn't actually interact with the cells correctly, or the insulin is fine, but the lock doesn't work properly, so your cells aren't responding to insulin correctly. And those might sound similar, but they do have different roles, really. So it all comes back down to insulin. But tonight I'm going to mainly talk about type 2 diabetes, simply by virtue of it representing 85 to 90% of all diabetes cases in Australia. So these are some Australia-specific statistics. About 1.7 million Australians are currently living with diabetes, or as of about 2018. And it's slightly higher for men, 6.1%, than for women, 4.6%. And now I want to take a second to just talk about some of the vascular complications of diabetes. And I've presented these statistics in a way that it really highlights how prevalent they are. So I want to really highlight, I haven't presented them like this to scare anybody because it can be a little bit confronting. But I think we really need to firstly recognize the prevalence of these complications and highlight the connection of these complications with diabetes to start understanding the problem and then addressing that problem. You need to understand the problem before we can start addressing it. So the first one is that people with diabetes may develop heart disease 10 to 15 years earlier than somebody without diabetes. And we can combine this statistic with the fact that heart disease is actually the leading cause of death in Australia at the moment. Not cancer or any other of those diseases, but heart disease. Secondly, every hour, 35 people with diabetes are hospitalised for heart or kidney disease. So we've mentioned heart disease, but kidney disease is another really commonly associated complication of diabetes. Diabetes is the leading cause of preventable blindness in Australia, and that goes for type 2 as well. Somebody did mention to me the other day that they thought it was only type 1 diabetes that could cause blindness, but it is in fact type 2 as well, and is the leading cause of preventable blindness. And this final statistic is one that I personally find the most shocking, which is that every 2 hours a lower limb is amputated, likely due to a chronic wound associated with diabetes. 1 every 2 hours. And again, I'm not trying to say this to scare anybody because it doesn't all have to be doom and gloom. On an individual level, there are many, many things that you can do to reduce your risk of developing these complications, and I'll walk through those right now. So the first thing, if there's anything you're worried about to do with diabetes or any of these vascular complications, you really want to start by seeing your doctor or any credentialed diabetes educator to discuss your individual risk factors. Everyone's different, everyone's got different genetic background, everyone's got different lifestyle, and all of these things are things that your doctor can discuss in detail with you to understand what your risk actually is of developing these complications. And some of the strategies they will likely recommend include maintaining a healthy weight, making healthy food choices, staying active, and a big one as well is stopping smoking. So all of these things can really contribute to significantly lowering your risk of developing these complications. And something else that, so if you're ever in a conversation with your doctor and you might just want to bring up any of these things and say, look, I'm worried about this or this, is to know your diabetes ABCs. So you can talk about how to manage your A1C, that's your HbA1c, or your glycated hemoglobin that will come up in a blood test, your blood pressure, and your cholesterol levels. So all those three things, A1C, blood pressure, cholesterol. If you're on top of those and you can manage those with your doctor, that will massively lower your chances of having a heart attack or a stroke or any of these other diabetic problems. So I just really want to highlight that on an individual level, it doesn't have to be, nobody has to panic at this stage. But these vascular complications are prevalent, and it's what I study. So let's go back to these main 4 complications of diabetes. We've got heart attacks, kidney disease, diabetic blindness, and chronic wounds. And each of these complications, we call them the vascular complications for a reason. It's because they all have one major thing in common. They all involve dysfunctional blood vessels. And let's delve into that in a little bit more detail for each of these. So starting with heart attacks, what actually causes a heart attack is the buildup of fatty plaque in the arteries of your heart. And when a person has diabetes, their coronary arteries are a little bit more susceptible to blockage with that plaque as a first thing. But secondly, the other thing we see in people with diabetes is that they don't exhibit enough growth of new blood vessels around the blockage in the heart. And I'll come back to that point a little bit more in a minute. But clearly there's a significant vascular component involved with this condition. Kidney disease, so your kidneys are full of little blood vessels whose job it is to filter things out of your blood. But when a person has diabetes, those blood vessels become damaged by the high blood sugar levels and they become leaky, so they let too much through, basically. Diabetic blindness, this is when the blood vessels in your eye become leaky. So if you've ever been to the optometrist and they shine a light into your eye and you can see all the blood vessels in the back of your eye is really highly vascularized, and when a person has diabetes, those blood vessels can become leaky, and that can be what leads to diabetic blindness. And finally, chronic wounds. So one of the main things that your body wants to do to heal a wound correctly is grow new blood vessels into the healing tissue to both clear things away, but also deliver new nutrients. It's one of the most important things that occurs during wounds healing. And in diabetes, unfortunately, we see that they don't grow enough new blood vessels into that healing tissue to deliver nutrients. So I think this really highlights that there's clearly a really significant vascular component to each of these complications. And that makes sense when you think about what insulin resistance actually means. So normally, insulin will signal to your cells to start taking up glucose from your bloodstream. They break it down for fuel, they're happy, everything's fine. But when your tissues outside this blood vessel here are insulin resistant, that means that the blood glucose doesn't get taken up and it remains circulating in your bloodstream. And when you think about the cells that are going to be in closest proximity to those damaging high blood sugar levels, it's the cells that make up your blood vessels. So to me, this is a really clear connection that helps us understand why there's such a significant vascular part of diabetes. And in particular, the cells that really bear the brunt of these damaging effects are this layer here, the endothelium, which is a single cell layer, so it's really, really tiny, of endothelial cells. And they're very specialized cells, and they sort of have 2 very important jobs. So these are the cells that my project mainly focuses on. Their first main role is in barrier function. So they're sort of the first line between what's in your bloodstream and what gets through to the rest of your tissues. And there's sort of 2 ways that things can get from your bloodstream out into your tissues. Either things can sort of wriggle through these tiny gaps between the cells, or things can be actively imported into the endothelial cells and then actively exported out towards your tissues. I think you probably already see that in insulin-resistant people, you're going to run into a bit of an issue here when this part doesn't occur because your tissues aren't taking up glucose. What this basically means is that your endothelial cells can actually start to accumulate glucose. And when there's a really high amount coming through here, they can start to accumulate quite a lot. You might be thinking, well, if the tissues can be insulin resistant and sort of decide not to take up glucose, why can't the endothelial cells? Well, that's simply because they don't need insulin to take up glucose. They're one of a few very special cells that can just take it up without needing to have responded to the signal that insulin sends them. And normally this is fine because their main job is to pass things from the bloodstream to the other tissues, so it makes sense that they should just be able to do that quite passively. But it's in these sorts of situations where that becomes a bit of a problem. But first, before I talk about those damaging effects of high glucose, I want to talk about the second main role of endothelial cells, and this is the one that I'm most interested in, which is in new blood vessel growth, or angiogenesis. So normally, for most of your life, in most healthy situations, there's not a lot of need for new blood vessel growth. Your vasculature develops quite early on during embryonic development, and it stays basically the same for most of your life. But there are some very specific situations where new blood vessel growth is really important. And I can tie this back to two of these vascular complications, heart attacks and chronic wounds. So let's just take a second to recap what actually happens in a heart attack. So this is your heart. You've got these two nice coronary arteries here that are supplying the majority of your myocardium with oxygen. When we run into a problem is where your coronary arteries actually start to develop plaque. So they're starting to build up these little fatty deposits that actually start to block the flow of blood through them, meaning that this downstream tissue here starts to not receive enough oxygen. And if that plaque gets big enough, or if a blood clot comes along and actually blocks that artery up completely, this heart muscle down here will start to receive no oxygen at all, and it will start to die. And that dying heart tissue is what causes the pain that signifies a heart attack. So this is what happens when somebody has a heart attack. Ideally, the body would very much like to avoid having a heart attack, so there are some things that it tries to do to help these situations. Basically, as soon as this heart muscle down here starts to think, oh, actually, I'm not quite receiving enough oxygen, it can start to send little signals up. I think it might be better. It starts to send signals up to these healthy bits of tissue to say, hang on, can we get some more blood vessels down here? Can we start, re-oxygenating and revascularizing this tissue? So the body will start to try and grow new blood vessels around that blockage to re-oxygenate that heart muscle. And I'll get back to that exact process of angiogenesis in a moment. But first I want to compare this to what happens in chronic wounds. So if you think about what a wound is, it's an area of damaged tissue, and not just damaged tissue, but also damaged blood vessels. If that tissue was vascularized, it clearly no longer is. And I mentioned before, but those two really important things you want to do to heal a wound are, firstly, you want to clear away any debris, bacteria or germs that might have gotten in when the skin was broken. But you also want to be able to deliver new nutrients and oxygen to the healing tissue as well. And both of those jobs are done by new blood vessels. So the first thing you want to do to try and heal up a wound is grow new blood vessels into that low oxygen healing area. But how does this actually happen? This is a pretty interesting process. So this process of angiogenesis is really what my project is all about. So I think it is just fascinating. In a normal artery, if you remember, your endothelium or your endothelial cells they're just lining the artery. They're sitting pretty quietly, not really doing much. But when they receive or when they sense a molecular signal that's coming from one of those low oxygen environments, it might be a piece of heart muscle or it might be a chronic wound, the cells sort of start to wake up. And you can see this one here is clearly stretching out towards that signal that's telling him there's somewhere out there that doesn't have enough oxygen. They start to divide and migrate, and they really just travel out towards that low oxygen environment, and they basically lay down a new blood vessel as they go. So they're literally wriggling out into the low oxygen environment. And over time, as that blood vessel matures, this happens relatively quickly, a new blood vessel will be, a mature blood vessel will be formed, and it can then deliver oxygen to the tissue that needed it. And that's angiogenesis. But if you remember, we're talking about our endothelial cells in diabetes, and they are sort of dealing with a lot. They're filling up with glucose. They can't send it to other tissues because of insulin resistance, and they're getting a little bit overwhelmed. So really one of the only things they can try to do in this situation is say, okay, we've got all this sugar here. We have to get rid of it somehow. Why don't we try to metabolize it? Why don't we try to break it down a little bit? So they commit some of this glucose to their metabolic pathways, so breaking it down to produce energy. And that sounds like a good idea, except our poor little endothelial cells are not really set up for that. They're not used to having to metabolize a lot of glucose. They don't really ever need to because they just sit happily on the inside of an artery. And we run into another problem with this plan, which is that some glucose breakdown actually requires a decent amount of oxygen. So let me just explain this little diagram here. So if this is a full molecule of glucose, there's this first step of breakdown which happens out here in this sort of general part of the cell. This bit produces a little bit of energy, but importantly, it doesn't require oxygen. So this part's fine. It doesn't do anything major. This part, however, is what occurs within the mitochondria. So you might have heard the mitochondria are the powerhouses of the cell. They're the little organelle within the cell that's mostly responsible for producing a lot of energy. So this is where the bulk of glucose breakdown happens, the most energy gets produced, and the most oxygen gets consumed. And this is a problem because if you think about why angiogenesis needed to occur in the first place, it's because there's a tissue somewhere out there where there already isn't enough oxygen. So in order for these endothelial cells to be trekking out into that low oxygen environment, what they really need to be doing is reducing their oxygen consumption. Sort of a cute little way we like to describe this is to think about your endothelial cells as your blood vessel builders. They've got their little hard hats on and their tools, and they are holding their breath. They're holding their breath because there's not enough oxygen around, and they're going to go out and build this new blood vessel. But when there's too much glucose around and they're just trying to clear it through, what this basically means is they lose their ability to hold their breath, and they can't adequately reduce their oxygen consumption in these conditions. So that's all really, that's a hypothesis up until this stage. We think, okay, the cells are exposed to high glucose, surely that means their metabolism goes a little faster, which means their oxygen consumption goes up. But we do actually need to, hop in the lab and test these things out. We can't just go off and say these things. So luckily, we've got some pretty good lab techniques we can use to actually study this, to study the effects of high glucose in these cells. So what we start off with, we can buy these from a company. We get the endothelial cells that have been taken from the inside of a coronary artery. So that's very relevant. These are the blood vessels that we're pretty much most interested in. So we've got some endothelial cells from the inside of these blood vessels. We can then put those cells into this cell culture plate. So this pink colour represents the cell media that we put on them. It's sort of a liquid food for them to keep them alive. So we put them in these plates, and then we can expose them to either normal or high glucose levels to see how that affects them. And then in order to sort of mimic the low oxygen environments in our body, we can then put that plate of cells into a 1% oxygen chamber. So that's very, very low amounts of oxygen that we use to simulate that. And then we can go on and we can measure how much oxygen are these cells consuming. How do they form tubular structures? So we have a special way of, in the lab, mimicking the way that these cells go on and form blood vessels. We call them tubular structures. And we can also measure how well they migrate towards a signal, which again is, so these are both very key angiogenic functions. So to start with oxygen consumption, the way we measure this, once we've done all those treatments on the cells, is we have this machine, and it's called a seahorse bioanalyzer of all things. Basically how that works is we put this little plate inside the machine, and it has all these tiny little probes that will come down and can actually measure the concentration of oxygen in the cell media. So we can track that over time and see, are the normal glucose cells or the high glucose cells consuming more oxygen? And this is a graph here that shows basically just that. So this darker bar here refers to cells that have been treated with normal glucose, and the lighter bar is high glucose. And then we've also got normal oxygen and low oxygen conditions. And what you can see pretty clearly is that when we expose those cells to high glucose levels, their oxygen consumption jumps by about 20%. So this really confirms our hypothesis that those high glucose levels are affecting the cell's ability to, ideally they should be reducing their oxygen consumption, but it is clearly going up under both of these normal and low oxygen conditions. Secondly, we want to measure tubule formation. So to do this, we start off with another little cell dish or a cell plate, and we fill it up with a gel matrix that basically works as a scaffold. So the cells can sort of settle into it and connect and behave however they're going to behave. So we put our cells into this gel matrix, and if they're happy and healthy, they will align, stretch out towards each other, migrate and divide a little bit, and form beautiful tubular structures. And that's what I've got images of here. That looks okay. So I'll just describe what's actually going on in these images, because I remember the first time I saw an image of this assay, I thought, okay, which part is the tube? But you can see, so this part here is sort of like a little clump of tiny little cells, and then this part is a cord or a tube that some cells have reached out towards each other, and they formed a beautiful little tube. You can see another one there, and all across that plate. So this looks beautiful. These cells are clearly pretty happy, when of course they are. They've been exposed to just normal glucose conditions. We can compare this to cells that were exposed to high glucose conditions, where, I mean, you can clearly see there's barely any nice tubes formed, and it even looks like some of the cells may have died. So from this we can say high glucose is really, it's affecting these cells' ability to form tubules. And then thirdly, we want to measure cell migration. And this is another very interesting lab assay that we can do. We start off with this chamber. So this is empty at the moment, this chamber. And it's at the bottom got this little membrane that has tiny little holes in it that cells can try to wriggle through if they're so inclined. And in the bottom we've got this liquid here, and this liquid actually contains that molecular signal that the cells will respond to. So the way this works is we put our cells into this top chamber, and if they're happy and healthy, some of them will migrate towards this molecular signal. We can then cut this membrane out. There'll be a bunch of little cells attached to it, and this is what that looks like. So all these little individual dots represent a cell that has successfully migrated through that membrane. And again, you can see under high glucose, many fewer cells are actually able to do that. So this is all very good data that we've obtained that gives us a lot of information about how high glucose is actually affecting our endothelial cells. But what are we going to do to help them? Because clearly they are having a bit of a rough time of it. And this, I'm going to describe a workflow that we would go through in order to identify targets to actually help these cells survive, because that's our ultimate goal. We want these cells to survive, so they'll build new blood vessels, so a wound will heal, or they can try to help prevent a heart attack. The first thing we want to do is analyze these processes that the cells are using to break down glucose. So if you remember, so there's this first stage, and then there's a second stage. There's a lot more to it than what I've put here. You know, this process involves many, many steps with a lot of different branching off points, and as does this part. The first thing we want to do is really analyze those processes and understand what's going on there. And then we want to think about, is there any point at which we can interrupt those processes? You know, is there an ideal spot where we can try and cut the cells off such that they can still produce a little bit of energy but aren't going to be consuming way too much oxygen? And then once you've identified a point that you can interrupt their metabolism, you want to try and carefully deliver a signal to the cells in order to get them to do that. Because all of this is happening in equilibrium. You know, there's a lot of different feedback that's happening, lots of different bits. So you want to try and do this pretty carefully. And this concept is called reprogramming metabolism. It's a relatively new field. There's a few groups working on this particular workflow in this field around the world. And I think it's really fascinating. And each group sort of comes up with their own little target that they think is going to be able to achieve a certain result. And it's a really interesting field to be working in. So we've gone through this process, and we've identified one enzyme which we think is going to be able to do the job. And its name is PDK4. Anyone who's ever heard me talk about my research is probably sick to death of hearing about PDK4. But it's a pretty cool little enzyme. It stands for pyruvate dehydrogenase kinase 4, and it's a mitochondrial enzyme. So this enzyme lives within the mitochondria for most of its little life. And its job really is to stop the mitochondria from processing glucose products any further. So basically it comes in at this point, it's within the mitochondria and can cut off mitochondrial metabolism. Ideally, this would reduce oxygen consumption, because if you can slow down mitochondrial metabolism, you're going to be able to reduce the amount of oxygen that the cells are consuming. So this is really ideal. We think this is going to be a really good target. And we have another piece of data here that I've collected that gives us a bit more information about the fact that this is a good target. So again, we've got our normal glucose, high glucose, normal glucose, high glucose, under normal and low oxygen conditions. And the first thing I want to point out is that under normal glucose, when we expose the cells to low oxygen conditions, PDK4 activity goes right up by quite a lot. So what this tells us is that PDK4 and its activity is really important for the cell's function in low oxygen conditions, which is perfect. The second thing is that we don't see an increase in PDK4 activity when we expose the cells to high glucose. So this tells me that high glucose is preventing PDK4 from doing its job when it should be going up in low oxygen. It simply does not. So these are two pieces of information that show us, yes, PDK4 is important, and yes, diabetes is having a negative impact upon it. So how are we actually going to increase PDK4? This is the carefully delivering a signal part of the project. We're studying this in two ways. Firstly, using HDL, or the good cholesterol, which I hadn't talked about before, but I'll explain all about that in a minute. And secondly, increasing PDK4 using a targeted gene therapy, which is very exciting and a very new area of research as well. So HDL. HDL stands for high density lipoproteins, and we vaguely consider it to be the good cholesterol. You know, if you go to the doctor and get your blood tested, they'll say, oh, your HDL cholesterol's gone up a little bit, that's good. Or it's gone down, that's not so good. But we can contrast that to your LDL, which they generally prefer to be a little bit lower. And that's just because while LDL is responsible, you know, these particles sort of roll around in your blood, LDL is responsible for delivering cholesterol to your blood vessels, which is what leads to the development of plaque, you know, fatty buildup in your arteries. HDL's job is in clearing that away, so its general function is presumably quite positive. We also know that higher HDL-C levels are protective against cardiovascular disease, and there's a number of reasons for why that could be. Firstly, because its main job is to actually remove cholesterol from vessel walls. But it's also been shown to have anti-inflammatory effects, and importantly, and of interest to us, HDL has been shown to increase new blood vessel growth. So that's very interesting. So again, we can test that out. We can figure out why that is. This time we've got our endothelial cells, we're adding high glucose, but we're also adding HDL. So this will let us ask the question, how does HDL treatment affect the way that these cells respond to high glucose levels? And the answer is pretty well. If we start off with our normal glucose cells here, they're doing fine, forming beautiful tubes, migrating plenty. Expose them to high glucose levels, and again, not as many tubes form, not as many cells migrate. But when we add HDL, I mean, this picture and this picture are practically identical. So what we can see is that HDL actually returns that level of tubule formation and migration basically back to the level or even a little bit higher than it was under normal glucose conditions, which is really exciting. But we do have to ask why that might be. Coincidentally, HDL increases PDK4 activity. It does a range of other effects that people have been studying for quite a while, but in particular, we've shown that it really strikingly increases PDK4 activity. So we can see normal glucose, high glucose, and again, we see that increase under normal glucose conditions with low oxygen exposure. Then when we expose the cells to high glucose, but we've added HDL to try and help them along, PDK4 activity, once again, is that induction of its activity under low oxygen conditions is restored, and it's significantly higher. So I should have mentioned earlier, these asterisks that we've added simply represent the degree of statistical significance. So we do statistical tests on this data to understand if it's a real result or if it's just by random chance. So this is a statistically significant increase. And very nicely, we also see that HDL decreases cellular oxygen consumption. So this is perfect. It both increases PDK4 activity, which if you remember is supposed to shut down the mitochondria, and we do in fact see that this reduces the oxygen consumption of the cells. So this is very promising, this data from HDL, but the next step always is to have a look at what's going on in preclinical models. So when I say preclinical models, what I really mean is animal studies. So I'm going to talk a little bit about that now, specifically talking about our work with HDL in diabetic wound healing. So we can conduct some pretty interesting animal studies using these technologies. And the first thing you want to do is you inject the mice with a drug that causes them to have high blood glucose levels. Then we can create 2 really tiny wounds and apply a treatment to try and help those little wounds close. Then we can measure the closure of those wounds, and also how well those wounds grow new blood vessels. We can use laser Doppler imaging to do this, which I'll explain in a minute. But first I really want to highlight that before we're allowed to do any of these animal studies, any of this preclinical work, the entire project needs to be really thoroughly inspected and approved by our Animal Ethics Committee. So they go through our project proposals with a fine-tooth comb to make sure that we're conducting these projects in an ethical way, which is something that's very important in medical research. And you're not even allowed into the facility unless you've done all these inductions. So this is something we take very seriously, is this ethical approval. So this is a graph that shows the wound closure in these animals over time. And I pretty much want you to focus on this red dotted line here, which represents diabetic animals that have been treated with, or their wound has had PBS applied to it. And PBS just means phosphate buffered saline, which is a fancy way of saying salt water. And it does basically nothing. So we use this as a negative control. So that's that red line there. And this blue line up here is the diabetic animals whose wounds have been treated with HDL. And you can see their percent wound closure over time is much higher than the diabetic animals without HDL. So we can say that daily topical HDL treatment significantly enhances wound closure in diabetic mice. So this was a very exciting result to achieve. But then I want to talk about measuring wound blood vessels. So this is an image that we would obtain from a laser Doppler perfusion imaging study. So basically a little laser, sort of like my laser pointer, will scan over the area, and it basically detects movement. So we want to make sure we've got our mice very, very still, and any sort of movement that we can measure, especially in this wound area, will be due to blood vessels and blood flow. So this blue area is this little sort of splint we put around the wound to protect the area, and then this circle represents the wound area. So you can see in the non-diabetic animals, there's not a lot of difference. They've both got a pretty normal amount of blood flow happening. But in the diabetic animals, especially, or specifically the PBS-treated wounds, the blood flow is much lower. There's only this one little area here, and the rest is actually quite blue. Compare that to the HDL wound, which is dark red. So low, or the blue area represents low blood flow, and red is high. So this is very interesting. So I'll just get my points on there. And you can see this represented in this graph as well, where this dark blue line represents your diabetic mice. It's about a 40% increase in wound blood flow reperfusion when we add HDL to these wounds. So again, a pretty exciting result. So that's the HDL part of this study. I'll just summarize that now. We've been able to show that HDL increases PDK4 activity, reduces endothelial cell oxygen consumption, it improves the endothelial cell function, as well as promoting new blood vessel growth in preclinical models. So overall, HDL is really showing quite a lot of promise at this stage as a potential therapy for diabetic complications. There's still quite a long way to go, but it's quite promising at the moment, which is exciting. So now I want to talk about the gene therapy approach. And gene therapies are a little bit complicated, but basically what it means is you've identified a gene or an enzyme that's dysfunctional, and you're going to add the gene for that protein or that enzyme to help it be produced. So in this case, what we're doing is we're supplementing production of PDK4 because we know that it's dysfunctional, or its activity isn't high enough in high glucose. We're basically going to add the gene for PDK4 in a very careful way and help it be produced a little more. But there's two very important things you want to think about when you're creating any sort of therapy, actually. Two specifically for gene therapy, but in general, what you really want to be able to do is achieve really targeted application of your therapy. You want to reduce off-target side effects as much as you can. So we need to be able to turn production of PDK4 on and off. That's an incredibly important point. And secondly, we need to be able to make sure it will only be produced in endothelial cells. And this is a particularly important point when we're talking about metabolism, especially within diabetes, because if you think about something like a muscle cell, it actually, that's one of the cells that becomes insulin resistant. We want those cells to be breaking down more glucose. They need it. They need heaps more energy and more fuel than an endothelial cell. So we really don't want to be messing with their metabolism in any specific, or certainly not in a potentially negative way. We really only want to be targeting endothelial cells. And we can do that. That's fine. Well, it's not easy, but we can do it. But you want to start off with the gene for PDK4. So this is literally the piece of DNA that if it's in your body, a little enzyme will come along, read it, and you'll get PDK4 enzyme produced. But in order to achieve this level of control, we have to add a couple of extra bits onto it. So we've got these two extra bits of DNA here, this blue bit and this orange bit, and I'll explain what each of those represent now. So this blue section sort of encodes for, or will be read to produce, a transcription factor that's responsive to an antibiotic. So when I say transcription factor, what I really mean is a specific little protein that's going to come along and sit on this gene and help it be produced. That's a factor that promotes transcription of the gene. And importantly, it's responsive to an antibiotic. So this is that bit where we can turn it on, or we can take the antibiotic away and turn it off. What that looks like is this transcription factor gets produced, we add the antibiotic, comes over here, you get PDK4 produced. Simple. And if we take that antibiotic away, that whole process would stop. But we've also added this orange piece here, which is, this bit's basically blocking this bit from getting produced. And this little piece of DNA is a sequence, a promoter sequence, that is specific to endothelial cells. So a promoter sequence is just a little small section of DNA that's going to let stuff come along and read the gene. It's something that other bits like to stick to, and once one thing sticks, the whole bunch will, and you'll get a much higher expression of your gene. And this bit is blocking this bit. So only things that exist in endothelial cells will be able to come along, stick to this bit, which means that only this bit, or this bit will only be produced in endothelial cells as well. So I hope I've explained that clearly, but these are those two levels of control that we can achieve. Endothelial cell specific, and we can turn it on and off with an antibiotic. So once you've been able to stick all these pieces of DNA together, that genetic sequence basically gets all packaged up, and you can then deliver it to a cell where it will start to produce PDK4 under these conditions of control. So where are we up to with that project? Well, we only really began creation of that gene therapy late last year. So I was actually working on this today in the lab. We're still working on being able to actually stick those pieces of DNA together in a way that is correct, and the way that we can tell that it is actually going to produce the product that we want it to produce. But we're making good progress on that so far, and we're hoping to begin testing in those preclinical models later this year. So that's a pretty exciting part of my project that I'm keen to get on with. So this is where we're at. We're at the creation of the gene delivery system stage. This next stage, this is the testing in preclinical models, has sort of two phases to it. The first thing you want to do when you create any therapy is make sure it's safe. We want to test it for toxicity in these animals. We want to test the dosage, so how much do we actually need to add. And we want to check the production of the enzyme as well. So how much PDK4 are we actually making? Is it enough? Is it not enough? And then we can go back and tweak the system a little bit. Once we're sure that it's safe and it's doing what we want it to do, then we can move on to being able to measure, does this increased PDK4 from this gene therapy increase blood vessel growth? Does it improve the cell's metabolism of glucose, or stop their metabolism of glucose at the required step? And most importantly, does it improve the complications that we're trying to model? So to conclude, basically I've been able to show you that we've identified two new ways of reprogramming metabolism of endothelial cells, using both HDL, which is a more holistic way of measuring it, of reprogramming the metabolism, and a really targeted gene therapy as well. This ideally will help our endothelial cells reduce their oxygen consumption, survive better in diabetes, and go on to grow new blood vessels. So all of this work, really, we're aiming to contribute to the creation of new therapies for diabetic vascular complications. That's our ultimate goal. And really, we want to be able to increase wound healing in these patients, prevent heart attacks before they happen, and all of that really contributes to significantly reducing the diabetes-associated mortality in Australia, which is a really important program, and I'm really glad to be working in this space. So with that, I would really like to thank my everyone from the Vascular Research Centre, but in particular my two supervisors, Associate Professor Christina Bursell, you can see here, and Dr. Joanne Tan. They're the ones that run this research program. It was Chris's idea to start with, so I'm really grateful to be involved with the project. And thanks to everyone else at the VRC as well. And thank you all for listening.
Thank you very much, Claire. What a fantastic talk. Okay, so I'd like to invite Claire to remain on stage while we go through some questions. So for those of you in the audience, we have Liz roaming around with the microphone, so please wait for her to come to you if you've got a question before you ask it. For those of you who are listening online, you can enter your questions into the Q&A section on your browser there. So to start off with, we're just going to start with one of the pre-submitted questions that was from Liz. And so Liz asks, is the damage to the vascular system that type 2 diabetes causes reversible?
That's a really good question. It sort of goes in two parts. So when you say the vascular damage of type 2 diabetes, obviously you're referring to, say, if somebody has a heart attack or if they have a chronic wound. And at the moment there are, for example, a chronic wound, there aren't many treatments available. So in terms of the vascular damage done by those complications, there's certainly things you can do to reduce your risk, but it still can have a significant effect on your life post that. But pre-vascular complications, type 2 diabetes, we believe, can be reversed. Actually, my granddad said this to me the other day. He said, my doctor said that my type 2 diabetes has been reversed. So that was fantastic news. He doesn't have to take metformin anymore. So what we basically think happens is when people can control those lifestyle factors like their diet, or they can exercise, or they stop smoking, what can happen is their control of their blood glucose levels can be improved by going through those things, such to the point where they no longer need to take insulin or a glycemic control drug like metformin. The important thing to note is that it's not a cure. So while you might be able to, if you've made some healthier life choices along those lines, you can go without metformin, but there is always the risk that it will develop if those changes reverse. So it's an interesting question.
Great. And I'll open it up to the audience now. Does anyone have any questions for Claire? Okay, while everyone is warming up here, we'll go back to one of our pre-submitted questions. Okay, so when a patient has a diabetic foot ulcer, what's currently being done in the clinic to treat and manage those wounds?
Yeah, so I mentioned before there's not a lot that can be done specifically to promote healing. So at the moment, if somebody goes into the clinic and they've got a diabetic foot ulcer, the main things that the clinicians will do is make sure it's clean, make sure it's not infected, they'll make sure it's bandaged well, and make sure that the patient isn't going to be walking on that foot. So it's all just sort of preventative things to try and help the wound heal on its own. They might remove some of the dead tissue from around it. But at this stage in the clinic, there's nothing that people are routinely applying to the wound that has been shown to be able to help it heal. So that's really the point where we're hoping to intervene with something like an HDL therapeutic or a gene therapy. Is at that early stage of the wound healing process where you really want to be helping the wound heal actively before it gets infected or before it gets to the point where somebody needs an amputation. So at the moment, really only preventative measures, but we're hoping to change that.
Great. Okay, so another pre-submitted question here. So you've discussed heart attacks and chronic wounds, but can these findings about endothelial cells be applied to any of the other diabetic complications like kidney disease or diabetic blindness?
Yeah, right. So I did mention kidney disease and diabetic blindness earlier on, and you might remember that I mentioned it's to do with leakiness of the blood vessels, which actually is a separate issue that involves inflammatory signaling and other factors like that. So the therapeutic development we're doing is really targeted at new blood vessel growth or inadequacies in that process rather than the function of the blood vessels and their leakiness. So while some of the work we're doing is valuable in its knowledge creation, so it will help us understand generally how endothelial cells are functioning and more like quite generally what diabetes is doing to those blood vessels. But the therapies we're developing are really only applicable to situations where not enough new blood vessel growth is occurring.
Great. Okay, so we have an online question here from Ashley, and Ashley says, I'm 63. Any of these approaches that you mentioned likely to occur in my lifetime?
I hope so. I very much hope so. That's always a tricky question to ask, or to answer certainly, because the process, we want to do it properly. We can't be rushing things to clinic if they're not safe. The COVID vaccine might have jumped to people's minds. That's a particularly special case because what we saw worldwide was an enormous amount of money, an enormous amount of cooperation between research groups to really work together to make sure that was effective and safe. And it was also the main priority of the regulatory groups, so they didn't have to wait in line. It just got straight through all those really rigorous processes. Unfortunately, that's not the normal situation. Researchers have to spend a lot of time trying to get funding, trying to make sure that we can go through all of these important regulatory steps in line behind whoever else has gotten to the situation before us. So unfortunately, it can take a little while, but it is important that we take the time to do it. And for this specific project, I'm the only person working on this specific aspect of it, so that can make it more difficult as well. But if we had a little bit more funding and a little bit more time to just spend working on it with more people, it would happen a lot faster, but I hope we get there in time for it to be useful.
So there we go. Any government officials listening, more funding is the the outcome from that. So another online question here from Kim. So Kim asks, for wound healing trial, was HDL administered topically or systematically?
Yeah. It was applied topically, and that's a really important distinction to make because there have actually been quite a number of trials now that focused on raising your HDL. So cardiologists made this connection years and years ago that higher HDL levels were better. And so we thought, oh, if we can just raise people's HDL levels, surely it will solve all the world's problems. Unfortunately, it didn't turn out to be quite that simple because we're now learning there's a lot of issues to do with HDL functionality, the degree of the disease that you're trying to treat. So systemic infusion of HDL has been tested quite a lot, but we're specifically applying it topically. And this makes sure that it specifically can interact with all the wound cells, it can really get in there, it soaks in. We've also seen that it actually seems to reduce the wound infection as well, which is just another interesting point. But yeah, topically applied.
Great. And another online question here, this time from John. So John asks, what is the maximum level of glucose that is recommended to prevent the suite of vascular problems that you've been discussing tonight?
That's a really good question. I'm actually not 100% sure of the exact level. We do measure it we measure the blood glucose of our mice. And we consider 15 millimoles per litre to anywhere above that we consider them to be diabetic, but I couldn't confidently say exactly how that, just off the top of my head, how that relates to humans.
No worries.
Nigel, we have a question.
We have a question. Excellent. Where are you, Liz? Oh, there. Okay, there we go.
Hi, just a quick question. How long does heart tissue death actually take to occur?
Yeah, that's a really interesting one. I think it varies between people. It doesn't actually, the tissue death doesn't actually start to occur until the oxygen deprivation is quite severe. So until the plaque has pretty much occluded or blocked up the vessel to a significant degree, or if a blood clot has come and formed in that area as well. So I couldn't say exactly in terms of minutes, but it takes quite a while for the plaque to develop. I mean, people can start developing plaque in their arteries in their 40s and then not have a cardiac event until their 60s or 70s, so it can be a 20-year process. But the exact death of the heart muscle, I couldn't exactly say, I'm sorry.
Okay, great. So another pre-submitted question here. So are gene therapies a common treatment strategy for diseases? And can you give us an example of some that are currently in use?
I can, and no, it's not. I think the first official gene therapy that was approved by the FDA was in 2017, so really, really recently. And that's because of all those safety issues. It's genetic material, you do have to be really careful with it. So 2017 was when that first started. And I think that was CAR T cell therapy. So CAR-T is chimeric antigen receptor T cell therapy, and it's basically when it's for cancer. You can get some of your white blood cells, and then they genetically modify them and reinfuse them so that they can better recognize the tumor cells and clear them from your body. That's a really exciting gene therapy, actually, but it is a really new area, and reasonably so for the safety considerations. But yeah, that's one example.
Great.
Actually, I just want to ask a quick question. From what I understand, you're using the PTK4 enzyme to stop the mitochondria from breaking down the glucose. So if the glucose isn't being broken down in endothelial cells, will that cause other potential problems?
Well, that's an interesting question. So the point at which we're interrupting the process, we don't believe so. Like I mentioned, that upstream process that's before the point we're going to interrupt it at, it should be fine to just churn through that glucose at that level. It remains to be seen, of course. I'm just hypothesizing. We hope not, but because that upstream process is relatively harmless, there are some branching off pathways that potentially could be something we need to keep an eye on. But we can assess all of that sort of thing when we do the pilot study and when we check the toxicity and all of those things. But that's a really good question.
Okay.
Hi. I was just wondering at what rate the blood vessels grow under angiogenesis.
Yeah, that's a really good question. Well, and I can't say exactly in the body. I think it really varies depending on the situation. But when we put our cells into that gel matrix, it takes about 6 hours for them to grow those little tubular structures. So 6 hours in vitro, or in cells, as we say, in the lab. But I couldn't say in the body. I think it really varies depending on the extent of the oxygen or deprivation that's happening. Whether a person has diabetes or not, there are a lot of different factors that could affect it, but that's an interesting question, yeah.
Okay, great. So we've got another online question for you. This one's from Yelena. So Yelena asks, I guess, whether any of these therapies would be useful for hypoglycemia and hyperglycemia related to meals.
Yeah, okay, that's interesting. I hadn't thought about that. We're so focused on preventing the effects of hyperglycemia that the hypoglycemia doesn't really factor in so much. But that's really interesting. I'm not sure. Certainly, maybe not so much for endothelial cells, but reprogramming the metabolism of your liver cells or your muscle cells could certainly be a strategy there. I mean, if somebody's hypoglycemic, you really want to make sure that your liver and muscle cells are responding appropriately. Or even your pancreas cells. If we have a look at exactly what's going on with your pancreatic islet cells, the ones that are responsible for producing insulin, I'm sure there would be something interesting we could use as a target in there as well.
Great. Are there any last questions from the audience? Okay, well, please join me once again in thanking Clear Primer for a fantastic presentation this evening. And thank you to you, our audience, as well, both for attending live here and for those of you that viewed online as well. So this has been our third Research Tuesday for 2021, and on behalf of the Research Tuesday team and the university, I want to thank you all for your time tonight. We're extremely excited to be back delivering these presentations on campus, and also to those of you that are viewing at home. We look forward to inviting you back in May, as you'll hear from representatives from the South Australian Law Reform Institute on the reform of the provocation law. Be sure to sign up for our mailing list to receive the latest information about that and all other research-related news. So thank you once again for tuning in tonight and for attending, and we hope to see you next time. Good night.